this is the overview of protein synthesis. Protein synthesis is the process of linking together amino acids to form polypeptide chains under the direction in the information of DNA. It is broken down into two parts, transcription and translation. Transcription is the synthesis of RNA under the direction of DNA. It produces messenger RNA or mRNA, and it takes place in the nucleus. And translation is the actual synthesis of the polypeptide chains, and it takes place in the ribosomes, and it's under the direction of mRNA. So ultimately, a living cell goes through protein synthesis to make proteins such as enzymes.
Okay, so transcription is the synthesis of RNA under the direction of DNA, and the purpose is to produce messenger RNA. And so there's three stages of transcription, and the first one is initiation, and it's regulated by transcription factors that attach to the promoter region. And then there's elongation, and elongation uses sense DNA strands, and the RNA polymerase II, it adds the nucleotides to the growing mRNA, and then that gets you to termination. And in termination, RNA transcript releases the polymerase and it detaches from the DNA.
This is initiation of transcription, and initiation of transcription is regulated by transcription factors that attach to the promoter region. And a transcription initiation complex is formed when transcription factors attach to the promoter region, which often includes the TATA box or 5' TATA AAA 3'. Then the RNA polymerase II attaches to the promoter like that, and the promoter is usually about 30 nucleotides upstream from the start of the actual gene. And after the RNA polymerase attaches to the promoter, the DNA strand starts to unwind, and the polymerase initiates RNA synthesis at the start of the DNA template.
Elongation of transcription begins as RNA polymerase moves along the DNA. It continues to untwist the double helix, exposing about 10 to 20 bases in order to pair with the RNA nucleotides according to base pairing rules. RNA polymerase II reads the sense strand, or known as the template strand, and adds about 60 nucleotides a second to the growing mRNA in the 5' to 3' direction. The non-template strand is not transcribed by RNA polymerase II.
Termination of transcription occurs when RNA polymerase II reaches a DNA stop region or termination sequences AAU, AAA in the DNA. RNA polymerase then continues 10 to 30 bases past the sequence and then releases pre-mRNA. Then the RNA is removed from the DNA and the mRNA is peeled from the sense DNA.
So mRNA processing must occur before the pre-mRNA leaves the nucleus. So first, a 5' modified guanine cap is added to the 5' end of the mRNA, and a 3' poly A tail is added to the 3' end of the mRNA, and these cap and tail are important for helping move the transcript out of the nucleus, and they also protect the mRNA strand from damage from the hydrolytic enzymes, and they're consisted of hundreds of adenine and guanine respectively, and they help ribosomes attach to the 5' end. And a mature mRNA strand at the end will contain a guanine cap, a leader region, a start codon, a coding segment, a stop codon, a trailer, and a poly A tail.
mRNA splicing is the process of removing introns from pre-mRNA, and pre-mRNA consists of exons and introns. It also consists of their snRNAs, which are also which are also known as SNRPs, and the SNRPs attach to each end of the intron, and the intron and other SNRPs pull at it to form the spliceosome, and exon 1 and exon 2 then detach from the intron to form the mature mRNA.
Okay, so mRNA coding dictionary determines which mRNA codon codes for which particular amino acid. And so how you use it is you take your first mRNA base from the 5', you get your second mRNA base, and then you take your third mRNA base from the 3', and you take those letters and you put them together to get your codon. And so the codon AUG, it codes for an amino acid, but it also determines the start of translation. And then when you have 3 codons together, they aren't an amino acid and they indicate a stop signal instead, and it's meant to mark the beginning of termination for translation.
So translation is the actual synthesis of a polypeptide chain, and it's under the direction of mRNA, and it's in the ribosomes. And so this is a ribosome, and these are the tRNA structures, and they all bring in different polypeptides to add onto the chain. And this is the mRNA strand, and these are the corresponding mRNA units.
Okay, so ribosomes are in charge of the specific couplings of tRNA anticodons with mRNA codons during protein synthesis. And so in a ribosome, there are 2 subunits, the large subunit and the small subunit. And each is made up of ribosomal RNA, which is made from the RNA polymerase 1 and other various proteins. And the ribosomal subunits are made in the nucleus of the nucleus. And so each ribosome has a binding site for mRNA and then for tRNA. And so in the P site, it holds the tRNA that carries the growing polypeptide chain. And then in the A site, it carries the tRNA with the next amino acid for the chain. And then the E site is the exit site, and that's where the discharged tRNAs leave.
This is a tRNA structure. tRNA molecules contain 80 nucleotides, and it folds together to form a 3-dimensional structure or rough L-shaped molecule. It includes a loop containing the anticodon and an attachment site located at the 3' side of the amino acid. And our tRNA is moved along our genes and is made in the nucleus by RNA polymerase 3. The anticodon is what contains the complementary RNA units.
Aminoacyl tRNA synthesis is a special enzyme inside a cell that connects proper tRNA to its proper amino acid. There are 20 different forms of the enzyme, each one for 20 different amino acids. Initiation translation begins in the 5' end of mRNA in the small ribosomal subunit at the start codon. Next, the initiator tRNA carrying methionine amino acid with anticodon UAC pairs with the start codon. Lastly, the large ribosomal subunit attaches to the complex. The arrival of this unit completes the complex. Initiator tRNA is in the P site, and the A site is available to the tRNA, bearing the next amino acid available.
So this is elongation of translation, and it consists of a 3-step cycle where each amino acid is added onto the preceding one. In this one, the A site is empty, so a tRNA structure can bring an amino acid to add to the growing peptide chain, and the anticodon at the end of the tRNA must be complementary to the mRNA codons to make sure that the correct units are being added. And after this process comes translocation, where the amino acid in the A site is given to the tRNAs in the P site, and eventually it moves over to the E site and leaves the ribosome. And translocation ensures that mRNA is being read from 5' to 3', and it keeps going until the whole polypeptide chain is complete, and it takes about 1/10 of a second to be completed each time.
Okay, so the last stage in translation is termination, and this happens when the ribosome reaches the stop codon in the mRNA. And so what happens is that the A site, instead of accepting a tRNA, it accepts a protein called a release factor. And so this release factor hydrolyzes the bond between the P site and the tRNA and the last amino acid in the polypeptide chain, which causes the polypeptide chain to be freed. And then the ribosomal subunits disassemble, and all the other components do the same. And then it's all reused.
This is an amino acid structure, and here's an amino acid. Each amino acid consists of an amino group, a carboxyl group, a central carbon, and an R side chain. Each amino acid is exactly the same except for the R side chain, because the R side chain is responsible for determining the 20 amino acids that it is within.
So this is peptide formation. A peptide is basically just amino acids bonded together, and in this, an ribosomal RNA acts as a ribozyme, which is an enzyme to bond the peptides through dehydration synthesis. It's called dehydration synthesis because water is removed as the bond is formed. And in the peptide, the carboxyl end of one amino acid bonds to the amino group of the next one.
Okay, so in order to get a polyribosome, you start with the ribosomal subunits. And so what happens is the ribosomes come together and they translate a single mRNA molecule. And so there's lots of different ribosomes translating this mRNA molecule all at the same time. And so this is forming a polyribosome. And so because of this, it allows the proteins, which are the polypeptides, to be formed quickly.
Levels of protein structure, there are 4 levels. So primary is the sequence of amino acid groups held together by a polypeptide chain, and it's determined by DNA. And secondary structure is loops and coils that are formed by hydrogen bonds between non-adjacent carboxyl and amino groups. The tertiary structure is a 3-dimensional form held together by the interactions between R groups, and then the quaternary structure is basically just primary, secondary, and tertiary all clumped into one form and structure.
This is post-translational modifications of proteins, and after translation, polypeptide chains undergo certain modifications, and this takes place in the Golgi apparatus. And here, sugars, lipids, and phosphates are added onto some of the amino acids, while other amino acids are completely removed. And proteins also undergo some modifications, and it affects their 3-dimensional structure.